methods were followed in the Human Genome Project for the analysis of human genome. One method is called expressed sequence tags, and the other method was called sequence annotation. Sequence annotation. In this method, expressed sequence tags, only the sequence of the coding regions the so-called template regions of the DNA were determined. Whereas in this method, sequence annotation, the sequence of nucleotides were determined both in coding and non-coding regions. So the entire sequence of DNA was determined, and the information obtained was later used for knowing the sequence of nitrogen bases. For this purpose, the DNA had to be fragmented. DNA of the human cell had to be fragmented, and it had to be coupled with DNA of other organisms such as bacteria and yeast. And the information obtained was analyzed using computers. In fact, the information obtained is so enormous that the database can be analyzed only by computers and can be stored only in computers, and this has led to the emergence of a new branch, interdisciplinary branch of biology, that is called bioinformatics. Finally, when the project was completed, the team of scientists working in various countries were able to come to some common conclusions that the human DNA has nearly 3.2 billion nitrogen bases. nitrogen base pairs. 3.2 billion nitrogen base pairs. Second important information available from this analysis said that only about 2% of this, only about 2% of this complete DNA is involved in functioning as genes. So just about 2% of the total DNA is capable of functioning as genes. About 28,000 genes have been identified. About 28,000 genes have been identified in the human DNA, in the human genome. Another information that was available was that approximately a gene may have about 3,000 base pairs. One particular gene called dystrophin gene dystrophin gene is known to be the longest gene, containing more than 1,000,000 base pairs. Chromosome 1 to 23, chromosome 1 has the highest number of genes, about 2,938 genes have been identified in chromosome 1. Y chromosome is known to have the lowest number of genes. lowest number of genes. Only about 231 genes have been identified in the Y chromosome. Its corresponding allele, X has more chromosomes than Y chromosome. So these are some of the important informations that have been available based on the studies conducted under the Human Genome Project.